आज ही डाउटनेट ऐप डाउनलोड करें। डाउटनेट पर होगा अब आपके सभी मैथ्स, केमिस्ट्री, फिजिक्स और बायोलॉजी डाउट्स का सफाया। बस अपने क्वेश्चन की फोटो खींचो, उसे क्रॉप करो और तुरंत वीडियो सॉल्यूशन पाओ। डाउनलोड नाउ।
In this question, first of all, they're asking how DNA fingerprinting is done. Now, we all know that DNA acts as our genetic material, and it is very important in all of the living organisms because it has a particular sequence of nucleotide bases, and it is according to this sequence that the proteins are synthesized. So the nucleotide bases code for proteins. Now, if we were to compare the DNA of two people, finding out the entire genome sequence would be a very tedious task. So DNA fingerprinting is a technique which was developed by Alec Jeffreys, where rather than comparing the entire genome, rather than finding out the sequence of all of the 3 into 10 raised to the power 9 nucleotide bases, what we have to do is we depend upon some variations in the DNA. So we find that there are some variations at genetic level which are probably created due to mutations. So such inheritable mutations are found at various sites in the DNA, because if we look at the genetic material, 99.9% of the nucleotide bases are exactly the same for all of the people. So people differ from each other in various characteristics only because of 0.1% of the DNA. And even in this DNA, there are many repetitive sequences. There are many stretches of DNA which repeat many times, and they do not code for any protein. So when there are some kinds of variations, some kinds of mutations in such repetitive stretches of DNA, and they are found in the human population at a high frequency, then we say that there is a polymorphism in DNA, and it is such polymorphisms which are the basis of the DNA fingerprinting technique, because these are inheritable mutations, so they can be passed on from one generation to the next, from the parent to the progeny. And therefore, by comparing these DNA polymorphisms, we can find out if two individuals are closely related, or we can compare the DNA of two individuals. So how is DNA fingerprinting done? We compare these inheritable mutations or these DNA polymorphisms of two persons. So what we are going to do? First of all, we have to isolate their DNA. Then we are going to digest their DNA with restriction enzymes, which are the molecular scissors, and they are capable of making cuts within the DNA at specific location. Now that we've broken down the DNA into fragments, we separate the fragments by gel electrophoresis technique. Then what we are going to do? After obtaining these desirable fragments, we are going to blot these fragments. That means we are going to transfer the DNA fragments to a synthetic medium such as nitrocellulose or nylon, and finally by using a satellite DNA as a probe, we are going to allow this DNA fragment to hybridize with the probe, and after hybridization is done, we can view the hybridized DNA by autoradiography techniques. So this helps us to find out about the kinds of polymorphisms, about the kind of inheritable mutations that are present in this DNA. And we can compare the genetic material, we can compare the DNA of two people without knowing the sequence of the entire genome, of the entire DNA. So next they are asking, we have to name 2 types of human samples which can be used for DNA fingerprinting. So since the DNA in every cell of our body is the same, we can use any kind of cell. We can use a hair follicle, we can use a cell from the blood, the bone, the saliva, even the sperm of a person. So any of these body parts can be a source of DNA, which can be used for DNA fingerprinting for identification. And they have asked us to explain the process sequentially. So we've already discussed the various steps. We isolate the DNA. Then we break it down into fragments by digesting it with restriction enzymes. Next, we separate the fragments using gel electrophoresis technique. Then we are going to transfer these DNA fragments to synthetic medium. Then using a satellite DNA as a probe, we are going to allow our DNA to hybridize with the probe. And finally, by autoradiography technique, we can view the hybridized DNA and compare two pieces or two samples of DNA.
क्लास 6 से 12 से लेके नीट IIT जेईई मेंस और एडवांस के लेवल तक 10 मिलियन से ज़्यादा स्टूडेंट्स का भरोसा। आज ही डाउनलोड करें डाउटनेट ऐप या व्हाट्सएप कीजिए अपने डाउट्स 8400 400 400 पर।